Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 2. Now focus on the first their instance of the construction of artificial recombinant DNA molecule. Artificial DNA means nothing but is what? Recombinant DNA, that is called as an RDNA, recombinant DNA. The construction of the first recombinant DNA that emerged from as in a possibility of linking of encoding antibiotic resistant with as in a native plasmid. So here mostly when LNDNA it is in a vector DNA. So when vector DNA, so in the LN piece, any one particular the LN piece to get add here, what happened in that ORI site, that ORI site to get add with as any one particular the DNA, it is completely autosomally they are replicating the circular extra chromosomal DNA, that is Salmonella typhimurium. Is for example it is a Salmonella typhimurium, it is in a E. coli. See, the Stanley Cohen and Herbert Boyer accomplished in 1972 isolated as in a their antibiotic resistant gene with cutting with help of the DNA plasmid with as in the responsible for as in the conferring antibiotic resistance. So in these two scientists to develop as in the new DNA. In these both scientists they get developed as a new DNA, means one LNDNA, one is in vector antibiotic resistant gene to get add there with help of the biotechnology tools. They developed as in the new DNA. They developed as in the new DNA, that new DNA, that is RDNA. Developed as in what? That is in a RDNA in 1972. The scientist was whom? They are Stanley Cohen and then completed Herbert Boyer. Next, after the cutting of the DNA at specific locations, because there became possibility with as in the discovery of so-called as discovery of the so-called as in a molecular scissors restriction enzymes. So the any DNA, for example, when we take as any one particular DNA, we cut with as any specific locations with the help of what? With the help of the any molecular scissors. We cut the DNA with the help of molecular scissors. When we are in these regions get using as any, the enzyme is what? That is in a restriction enzymes to get restricted. They get complete to get restricted. The cut the piece of the DNA into linked with as in the plasmid DNA. So when for example it is in a one DNA, it is another DNA. Okay, so when we are cut here in this piece and in this piece, we get joined here to develop as what? New DNA fragment. To develop as a new DNA. This plasmid DNA, they get act as a vector to transfer it as a piece of DNA to attach to it. So this DNA to get easily to get transferred into as any other host organism. Transfer into as any host organism. Here we are acting as what? It get act as a vector. It act as what? Vector. Next, you probably know as in a mosquito that get act as in a the insect vector to transfer the Malarian parasite to human body. So the mosquito also they get act as in that mediator, right? So they get Malarian parasite to get carried to as in into as in a human body. In the same way, the plasmid can be used as a vector. So here plasmid also get act as in what? Vector to deliver of alien piece of DNA into as a host organism. So we are directly to not injected to as a into host organism. We are required for as a one mediator. That mediator to get easily to get produced as in multiple copies on the host body. Next, the linking of the antibiotic resistant genes with the plasmid vector become possible with as an enzyme is DNA ligase. For example, when we are cutting this DNA is there, am I right? So this DNA, small DNAs are there here, am I right? So after cutting off this DNA, the small DNAs is there. In these small DNAs, how do we are completed? They get attached here with help of the here one required one enzyme. That enzyme is what? DNA ligase enzyme is required. So the number of the DNA fragments to get attached with help of the enzyme is what? DNA ligase enzyme. DNA ligase enzyme to get help. Next, see, mostly which get acts as which acts cut DNA molecule in joints and their end. So the mostly the ends of the regions we get completely to get attached to as an a the DNA. This makes as an a new combination of the circular the autonomously the replicating DNA created by as an a in vitro is known as in the recombinant DNA. So in this process based on we developing as in the new DNA. In this total process based on we are completely developed as a new DNA. This DNA is called as what? RDNA. That is what? RDNA, recombinant DNA. Let us, when this DNA is transferred into Escherichia coli, it is in Salmonella. Here another vector here, E. coli is present. E. coli is present. So, in the Salmonella is transferred into as an E. coli now. The RDNA they're completely see in this E. coli in this E. coli the salmonella DNA get transferred into as an E. coli cell. Here what happened? The bacterium is closely here the bacterium is closely related to as in salmonella could be replicates using as in a new host DNA polymerase enzymes and is make as in a multiple copies. So in the salmonella DNA as well as Escherichia coli DNA is mostly they are similar. So then what happened? Their E. coli bacteria they think about as in it's my own DNA. Then they start the replication with help of in a polymerase enzyme. It is get produced as in a multiple copies. The ability to multiply copies antibiotic resistant gene in is E. coli, it was called as in cloning the antibiotic resistant in a E. coli. So then what happened in the cloning process? Automatically the antibiotic resistant genes we developed as in a E. coli cell. Okay. Next, you can hence infer that there are three basic steps in a genetically modified organisms. So when we developed in this genetically modified genes, we followed as in a the three steps mainly. The first one is what? The first one is the identification of the DNA with desirable genes. First one is what? To identify the DNA with the desirable genes. So important genes we are completely first to get identified. Second one, introduction of the identified DNA into host cell. So after the identification to identify as in a host cell also. And third one, maintenance of introduced DNA into host cell, transfer of the DNA to its progeny. So after to get maintained to with help of the vector only we get transferred into as a DNA into completely host to cell. Okay, now these three steps compulsory they get followed. Okay, it is in a basic introduction of in a biotechnology in principle. Okay, remaining we discuss in next class. Thank you for watching. Please share and subscribe. Thank you.